Recording in progress. Okay, good evening, everybody. I am so pleased that you are able to be with us here this evening at our first virtual animal science program of 2021. Tonight, you will be reviewing and seeing Roaming the Rumen. And so just as a way of kind of keeping track of how we run Zooms, make sure you have yourself muted, and you can certainly take off your video if you desire. It might help with bandwidth. And if you've got any questions as we're going through here tonight, make sure you put them in the chat button on the bottom. Try and keep your friendly banter back and forth kind of on the down low, if you would, and keep the chat box for the questions as we go here through this evening. So tonight is the first of this series through May, and next week or next month we will be doing a session on feeding and caring for swine without ractopamine. So if there's more sessions you'd like to attend, please go to the link in the bottom right, and that will get you to all of the sessions, or if you just want to go to 2 of the sessions, you can sign up for as many as you'd like. If you would like to receive some educational credits for this evening, the link is here, and please fill that out as you're going through the session tonight to gain some of the learned objectives or learned things that you've seen from our speakers or what you hear from the speakers this evening, and that will help you as you work through your project. Please send this to your county fair or your livestock committee. Here is the way you can keep up with Wisconsin Youth Livestock Program. The website, the YouTube channel where all of these recordings will reside if you want to go back and check them out. And also if you're on social media, please keep track of us in that way. All right, and this evening we will end with a evaluation. So please make sure you fill it out for us. This is a great way for us to be able to put on programs with topics that interest you and at times of the day that might interest you. So please, without a hesitation, make sure this evening before you end that you fill out that evaluation form. All right, so we are pleased to have our speakers here this evening to kind of go through Roaming the Rumen. And I'm gonna stop share here and turn on the video for our speakers. And so we have Eric Ronk with us, who will be talking a lot here this evening, and Luiz, Dr. Luiz, who will talk a bit more about the research side of things. So at this point, Eric, if you want to go ahead and take it away, we will go ahead and begin with you.
Okay, thank you, Bernie. Can you hear me okay? Okay, good to hear. Well, good evening, everyone. My name is Eric Ronk, and I work as an assistant faculty associate in the Department of Animal and Dairy Sciences here at UW-Madison. Been here for just over 4 years, and prior to that, I was in Calumet County as the agriculture agent. So I don't know if we have anyone from Calumet on tonight. Originally grew up just outside of Green Bay in Denmark, Wisconsin, on a small dairy farm. And so some of my responsibilities here at UW is teaching our Animal Science 101, kind of our first intro to animal science classes here for a lot of the undergraduates, and also some farm and industry short course classes, more on the dairy related side, and been involved in some feed efficiency research here as well. And so I even packed my special mask today with the cow print to coincide with our Roaming through the Rumen activity. And so we're going to kind of start off tonight just kind of going through some of the basics. So I think Bernie had included a kind of a handout with some basic information about digestion and thinking about the 4 compartments specifically of a ruminant animal. And so my background is more on the dairy side, but certainly similar across sheep and goats and beef cattle and dairy cattle as we kind of think of the basic digestion and how nutrients are absorbed in those animals. And so we do have today, or this evening, a special model, and I know I have to be very careful with it, or Sandy, our lab manager, will be really upset with me because this is several years old. It's had better days, but this is an actual live, or was a live a ruminant animal. I believe it was a steer, and we went through and preserved this. So you can see all the different compartments here. And so I'm going to kind of start off talking just generally about what ruminant animals can eat, because they can eat things that you and I wouldn't be able to get energy from. And so I'll show here, we have what we consider TMR, or total mixed ration. And I usually joke with tour groups that come here with the Dairy Cattle Center that this is like a casserole, or kind of a hot dish, that everything kind of gets mixed in. And I probably should pause a little bit here and kind of just give you a general overview of our facilities. So we are standing in the Dairy Cattle Center here. This is kind of our welcoming area when we get tour groups in. So unfortunately, with everything with the pandemic, it's been quite a while since we've had any group. But on campus, we have room for about 84 cows. It's a tie stall operation. And so it was remodeled about 6 years ago, I believe. They kind of did it in phases. And so they put in a milking parlor and extended some of the stalls. And really nice. We are literally about a block or two away from the animal science building, where Bernie and I both have our offices. And so really nice for students to be able to have access to cows from education, research, outreach, and everything. And actually right above us here is a classroom. And so a lot of times I will use that classroom for some of my classes, and we can go right down and do some lab activities. The other thing I should note is our largest dairy farm is about a half hour north of here, and that's Arlington, otherwise known as Blaine Dairy. So we have about 500 cows there. We have about 100 dry cows, and we raise a majority of our calves there until about 12 weeks of age. And then they go to our Marshfield location. And so they're raised there, and we have about 100 milking cows up there, and as well as the beef unit, sheep unit, as well as some of our areas and units that we're able to do research and have live animals for teaching and everything on campus. So certainly a great opportunity to have those. And so that's kind of the general outline of some of our animal units. And so I'll kind of come back here now. We will talk through some of the TMR. So that's pretty basic, what we're going to feed to an animal. On our dairy cattle side, a lot of these cows are formulated to have over 100 pounds of day that they're eating. So certainly a huge amount of feed that these animals are eating because of the amount of milk production that they're producing. We also have here on the top here, closest to my hand, is corn silage. So if you're familiar with cornfields, usually in August or September around Wisconsin, that's when we're going to go ahead and harvest corn silage, depending on moisture and everything with the kernels. So very key in a lot of ruminant animals, and especially in the dairy world. The next one we have here is our straw. And so as some of you might be familiar with using for bedding, pretty typical, but we will also actually feed it to some of our dry cows. So there's less energy in the straw, and certainly has some fiber. And we will definitely use that in some of our rations or diets. Then moving over to our last bin here, on the top, we have cottonseed. And so pretty typical to have that in a lot of our lactating cow diets. And then the bottom one there we have is our high moisture corn. And so we're going to harvest that a little bit differently than we do with corn silage, but certainly fermentable, a lot of starch there for animals to be able to utilize. So that's kind of typical on the side from ruminants, that they can eat forages and grasses and different things and be able to break that down. And so if you think of an animal, whether it's a beef cattle or a dairy cow, they're going to use their tongue and they're going to pull that feed in, and they're not going to chew it the best. Kind of like how maybe sometimes your parents tell you to chew your food a little better. They're not always the best with it. And so they'll actually swallow that, and it'll go into the largest compartment, the rumen. And so I have the esophagus here, which is about 2 to 3 feet long. It's not all here, but it'll work its way down through there and get into this large compartment known as the rumen, or the fermentation vat. So there's a lot of things from methane and CO2 and fermentation that's going on here. It's an anaerobic environment, and that animal is actually going to be moving that feed around. So if you watch a beef cow or dairy cow, you'll actually see it's on the left side especially. You'll see the movements that go on in the rumen area. And that animal is basically breaking that, trying to ferment that feed and be able to utilize some of those nutrients. And so you might be wondering why there's a hole here. And there's a specific reason for that. And our animals here on campus, with UW being a key research facility, we actually will insert cannulas. They're sometimes known as fistulated cannulas. So I have an example here, kind of looks like a disc type figure. And we will actually insert this into the cow. Usually they're second lactation or greater because they're still growing at that time point. But we will actually insert this into the cow, and we will, so this is exactly where you would see it placed later on in our demo tonight that we're going to be doing. And we have to work with a veterinarian to do this. And so a veterinarian will go through, it's a procedure, and they will cut, and we have to be very careful to follow up and make sure we clean and take care of that area. But once the animal goes through that procedure, they do really well with things. And so that's kind of the basic breakdown of things going on in the rumen. And then we kind of think of the next compartment. We have the reticulum area. And so that reticulum area is kind of a honeycomb type appearance. I actually have a picture here, if we can zoom in, of what it looks like. And so one of the key things that we sometimes talk about with the reticulum area is that sometimes little pieces of metal or nails or something can actually puncture that reticulum area. So one of the things we'll do from a management standpoint is put a magnet in an animal. And so we can actually insert this in, it'll kind of make its way down to that reticulum area and would attract any type of small pieces of metal to prevent injury for that animal. So one of the things that we do there with the reticulum area. So the feed will make its way to the reticulum, and then the next compartment it's going to make its way to is the omasum. And so the omasum is known as kind of that many plies, or almost looks like a book chapter. I think I have a picture here that I might be able to show. But in this area, the omasum, the kind of the main digestion that is happening is a lot of times with water. And so the feed is going to start to get a lot more dry at that point. So this is what it looks like with the omasum. So you can kind of see how there's those folds, and you can kind of see that there. Also, some of the electrolytes and different things can be absorbed across the omasum. And then we make our way into the abomasum, and that's kind of this last area here. And a lot of times we refer to this as kind of the true stomach of the animal. And so the pH is going to drop. If you think to some of your science classes, maybe some of you have had, it's going to be more of an acidic environment. And that helps with digestion and breakdown there in the abomasum. And so once it makes its way through the abomasum, we get into the small intestine area. And then that is about 120 to 150 feet long, depending on the animal. So it's way longer than the entire animal. It's pretty incredible. And that's because of the time that it's in there to be able to digest that material. And then we have the large intestine, and then kind of the back end of the animal, we have the manure or the feces. Whatever doesn't get digested will be used for hopefully fertilizer in that case. So that's kind of the general digestion and the different compartments of a ruminant animal. To talk more about on the research side and some of the other things that we have here on the table.
Yeah, thank you, Eric. And now that you all learned a lot about how cows digest feeds, I'll try to discuss a little bit with you from a research perspective. What do we do here at the department that's so important from the nutrition side? So basically, I'm assistant professor here, but the main focus of my research and extension programs is to learn about how lactating dairy cows digest and utilize different feeds, and how that can improve milk production and health. So this is one of the areas that we work the most, and obviously understanding the rumen, understanding factors that may affect the rumen is one of the most important things associated with that. Another important set of questions that we very often try to answer is how can we improve the quality of those different feeds? And we try to understand what are all the nutrients associated with different feeds. For example, Eric showed you corn silage, straw, and corn grain, how those different feed stuff, they differ in nutrient composition that may differ on how we formulate diets. And this is a little bit of what we try to do. But I don't want to get you bored today, so I'll try to share a little bit more of the fun things we get to do as a research scientist here at UW. So we'll try to describe a little bit more of the rumen. So Eric described to you earlier the rumen cannula and how the rumen cannula works. And yes, we use a lot of cannulated cows for research, but what a lot of people don't know is that the rumen cannula was originally designed for veterinary medicine. So for those of you that have some aspirations about veterinary medicine, there's a lot of important things about the cannula, because having a cannula that gives you access to a cow's rumen allow vets to collect rumen fluid from a healthy cow and transfer that rumen fluid to the rumen of a sick cow that maybe had some digestive disorders. And that helped a lot to help some of those cows to recover. Okay? So that was the main reason behind the development of this type of technique. Now, as a nutrition scientist, part of what we enjoy doing is understanding how digestible is a feed. And let's be honest, if we want to understand how well a dairy cow digests a feed, the best answer you can get is from a dairy cow. There's no comparison. But we often cannot feed a lot of dairy cows to properly learn that. And even though very often we test different diets, we had to develop some new techniques that allow us to measure that. So today we will talk about two different techniques, and they are very similar in nature. And the demonstration we will have a little bit later with the cannulated cow is one of those techniques. So the first thing I want to show you is what we call in vitro digestibility. So when you have cannulated cows, we can go to the barn, collect rumen fluid, and then with those rumen fluid, we can put that inside flasks. So if you are guessing what this is, yes, this is rumen fluid. One of our grad students, Ben, you'll meet Ben a little bit later during the cow demonstration, collect this rumen fluid a couple hours ago. So basically what we do is we collect rumen fluid, put in a flask like this, and then we have bags like this, which have different feeds. In this case, we have a corn grain sample, and we add the bag inside the rumen fluid, and we leave there for a given amount of hours. Later, we analyze the different nutrients that were remaining in this particular bag. So if you know the amount of nutrients that were originally in the feed ingredient and the amount of nutrients after the procedure, we know exactly how much it was digested. And obviously, in order to do that, we have a whole setting in our lab that has CO2 going into those flasks, because it's a fermentation chamber, an aerobic environment that needs CO2. We also keep that in a water bath set at approximately 100 degrees. Because if you put your hand inside the rumen of a dairy cow, yes, it is very warm, okay, because there's a lot of fermentation going on. The other thing that we typically do is we have some laundry bags here, and we have a different type of bags. So those are in situ bags. Here we have high moisture corn and corn silage samples. So sometimes we add those bags into the laundry bags. Then we go to the dairy cow, and Ben will show that live for you later today. Then we go with those samples there. We pass those through the cannula, and then we're gonna put inside the rumen, and we're gonna leave there for an X amount of hours, sometimes days. And then when we reach our time points, we go back, collect, analyze, and then we know exactly how well digested it was. So these are some of the things that, from a research perspective, we can run in our department. And for those of you that have interest joining our school as an undergrad or as a grad student, look look for us, because we always have interest having students joining us and learning about those different techniques. And I won't lie to you, I was a grad student here and I decided to come back because this is very likely the best research facility for dairy nutrition in the country. So for those of you that have this aspiration, keep that in mind. And I think that's the main research discussion we had for this part. I believe we're gonna move to the dairy cow next.
Just one quick question, Luiz. You might need to mute it. Okay. Just one head quick, what were some of the things at the bottom of the bag that why would they need to be there? What's the next step? Or maybe you'll hit that in the next step, but do you want to comment on that?
Sure, absolutely. So the reason why we have, these are some rubber weights. This one is not rubber, but these are some weights. And the reason for that is, as Eric described, a lot of times the cows consume a lot of material, but they don't really masticate a lot while they are consuming feed. So the way the dairy cow operates is they will eat a lot, then they will go rest. And while resting, they keep ruminating. So the rumen starts moving back and forth, throwing feed around. And this way they start a process where they regurgitate, chew a bolus, and consume that again. So during this process, the rumen moves a lot. So if we don't put some weight into those bags keep moving back and forth. And our goal is to have those bags sitting, if possible, in a more stable position to make sure we don't have a lot of variation in the digestibility. That's why we have those there.
Nice. So you talk a lot about dairy cows, because that's a large part of your research. How does this kind of work that you've described work in beef and other small ruminants like sheep and goats?
So this is actually very common for beef cows as well. Okay, there are many facilities across the country that have cannulated steers that are typically used for similar type of trials. In general, when we test a forage or a feedstuff, we can assume that the digestibility of a dairy cow is representative of a potential digestibility of a steer. Okay, not to formulate diets, but you have an idea on the quality of the ingredient. For small ruminants, we can also have cannulas. Obviously, they are much smaller than those cannulas, okay? And we can run similar process. A lot of what we know today about the rumen actually was discovered with trials with sheep. So a lot of the initial studies trying to understand the rumen, they were cannulated sheep. And then we start learning about those, translated that type of research towards beef and dairy cows, and continue from there. So regardless of the ruminant type you want to work with, I believe there is a lot to learn and a lot to do, right, Eric? So.
Is there any other questions?
I was just seeing on the chat some questions about the cannula, about how it maybe potentially get infection and everything else. And you might have to help me out, but I think that technology started like back in the 1930s originally is when they started figuring out this process. So it's been around for quite a while, how to go through it. And like I was saying, there's a veterinarian that goes through and we keep kind of a certain amount of cows in the dairy herd here to be able to do some of this research. And one of the other things we can do if we have a cow that is sick, right? We can actually take some of the rumen fluid, and if we're going to drench the cow that is sick, we can also include rumen fluid from a healthy cow in order to try to kickstart that animal's, to be able to help and get better by transferring that rumen fluid. So that's something that we can do. But as far as the cannula goes, when they go through the surgery, yes, we definitely have to clean. We check daily, do a lot of things. But once the animal kind of goes through that process, they do really well. I mean, once this cannula is inserted, they keep this cannula for the rest of their life. But once they're in, we have very good luck with it. It's not something that usually gets infected. We do have the manager who are some workers around here that will check on it. One of the things that can happen from time to time is with the cannula, there's actually a plug that goes in here in the middle. And sometimes that plug can kind of get pushed out. That's probably the most common problem that we see with the cannula. And so that's one thing that we just have to be very careful of and check our animals that have the cannula. And again, doing the cleaning and everything else when the animal goes through the surgery. But after that, they do really well. Sometimes we might replace this to put a little bit bigger one in, but for the most part, they do well. It's almost like having a window, right, to access their rumen.
Yeah, and it's important to remember that we go through multiple protocols in order to make sure, first, we are allowed to do surgery like this for cannulation. And in order to have that, we will have trained veterinarians that will come here, do that for us, and make sure they keep track of the animals to make sure that there is no issue with any of those cows. So it's not like we randomly choose a cow to do that. There is a lot of procedures behind that in order to make sure these animals, they are safe and they do not suffer anything. So, and just to complement what Eric said, after surgery, 1-2 hours after surgeries, those cows go back to the tie stall and eat normally. So it's important to also learn and remember that the way a dairy cow feels pain is completely different than a human. You know, the nerves and the entire system is very different. So it's a very safe procedure, and it's actually very, very important for us. So.
Great. I think I'll just make one comment and then we can go into the barn. So a lot of the comments tonight so far have been around dairy cows. I went to graduate school in Nebraska and other states that do a lot of work in like beef nutrition. And there were steers that were cannulated to do much of the same work that we kind of work for in the dairy cow part, but they were trying to figure out all the parts as it related to like feedlot work and how steers did feed efficiency research projects and other types of feed trials that are similar. So we just want to make sure you realize, because I know many of you are probably beef kids or maybe show other small animal or the sheep or the goat projects, or you may show poultry, and that's great too. This is all part about learning all different facets of how each animal works. And the only other thing I'd probably just mention is, and we get this question a lot, we hear that, you know, ruminants, we're talking about ruminants a lot tonight, but we hear that swine are monogastrics. Can you tell the students just a little bit about the difference between those digestive systems in just a really quick, easy way? And then we'll go to the barn.
Yeah, absolutely. Before I answer that question, there is just one clarification I'd like to make. Someone asked if we will watch a surgery. No, you're not going to watch a surgery. Believe me, it's virtually impossible to have a protocol in order to watch a surgery. So please don't expect us to do that. But going back to the question, the main difference between monogastric animals like yourself or ruminant animals like a dairy cow, beef cow, or small ruminants is the ruminant animal has one stomach with 4 compartments. And it's important to remember that one of those compartments is the rumen, which is a fermentation chamber. So basically ruminant animals, they ferment feeds first, and after that they start the digestion process in the abomasum, which is similar to our stomach, and the small intestine, which is exactly like our small intestine. Monogastric animals like humans, swine, poultry, particularly humans and swine in this case, the digestion starts with the saliva and then continues in our stomach. And this is a chemical digestion. So we have basically enzymes and acids like hydrochloric acid in our stomach that helps with the digestion process. And then when it reaches the small intestine, it's purely enzymatic. So basically the ruminant is a slightly more complete digestion because it gives us the potential of fermenting feeds that we cannot digest, like forages, for example. Humans can barely digest fiber, whereas ruminants can digest up to 50–60% of the fiber that they consume. So these are the two main differences between those.
Excellent. Well, there's more questions, but I think let's head to the barn in lieu of time. And I'm going to turn this over to Lori. I want to make sure I thank Lori. Lori Wick is the 4-H Positive Youth Development educator in Lafayette County. comes also to us with an agri-science teacher background. So I am going to put you guys in the healthy hands of her while we take about 3 minutes to go into the barn, and we'll show you where the cannulated cow is and just talk through all the parts and all the details in there. So hang tight with us and hang out with Lori while we get back.
Perfect. Thank you very much. Okay, we're going to do 2 quick questions. We are going to use your chat box. So we'd like you to go over to the chat box and in a couple of words, just a couple of words, tell us in the chat one thing that you have learned about nutrition and or ruminant animals tonight from the information that was given. So in a couple of words. And the second thing we're going to have you do in that same chat is on a scale from 1 to 5, 1 being not very important and 5 being very important, give us a number and tell us how important is nutrition to your specific livestock project. So you've got 2 questions in 1 chat. You are going to tell us one thing or a couple of things that you've learned tonight, and you're going to give us a number on a scale from 1 to 5. How important is nutrition to your livestock project? And while you guys are doing that, I am going to pull up a couple of poll questions that we are going to take a look at. So as you're getting done putting that information in the chat, thanks for doing that. It's going great. I'm getting ready to launch a couple of poll questions. We'll test your knowledge from what information you may have learned tonight. Awesome job, guys. And thank you for keeping your mics muted tonight. It's made the presentation really easier. Okay, we are going to launch a couple of poll questions for you. So I believe there's 5 questions. If you just take a minute and look at the screen, you can go ahead and answer the questions. The first question, it says, what's the most important nutrient for animals? So if you can find your poll button down there and start to answer. Again, I think there's 5 questions. Yep, 5 questions, and we'll go over the answers here quick as soon as we get everybody in. We have around 230 guests tonight, so we'll give it a couple of minutes here for everybody to get their poll questions in. That's great. I love reading what you're putting in the chats. I love seeing that you think nutrition is a very important part to your livestock project also. Oh, nice job. Okay, we are about 1/4 of the way done. People are starting to answer the poll questions. And Harper, I see you don't have something that you've really learned tonight, which is fine. Thanks for answering though, honestly. Okay, we've got about 130 out of 230, so we're still climbing. We're doing good. Okay, we are going to go ahead and just give a couple more seconds on the poll. We are just about at 160 out of 230. They are ready for us when we are done. So we're going to just go ahead and stop the poll right here. If you didn't get a chance to answer it, that's okay. We appreciate that. We're going to share results right quick so you can see what they are. So the answer to question number 1 was water, the most important nutrient. Number 2, how many compartments in the ruminant? 4. Number 3, which compartment is the storage compartment? That was the rumen. Number 4, which compartment is similar to a non-ruminant animal such as a pig? The abomasum. And then which compartment holds any hardware? That was the reticulum. So nice job, guys. Thank you very much. I'm going to turn it back to Bernie.
All right, everybody, we're back, and I'm just going to spotlight the video. So just hang tight to make sure everybody can see it. All right, so here's our cow. So we had to give you a little bit of the heads up related to what we were doing and about the stomach and all that intro stuff. So now you get to see the cannulated cow. And I'm just gonna turn this over to Eric to move through this. And if anybody, this has been way doing his PhD here at UW-Madison. All right, Eric.
All right, so hopefully you can all hear me yet now that we moved to our teaching arena area. But we have one of our cows here from the Dairy Cattle Center. So as we were explaining before, we have the cannula here, just like we were looking at previously. And this is that actual plug that I was referring to. And so you can kind of see some of the hardware here on the other side. Sometimes what we'll do is there'll be a clip that we can then clip to that. And then that way, when we put bags like this in, they don't go floating away on us, so we don't have to keep to dig in. I've had it happen before where the clip breaks and you got to go fishing for a little while, and that's not so fun. But if we want to go ahead, we can open her plug here. I will say it's probably going to be pretty full. She's been eating pretty well here, and so she they, like I was mentioning, they eat pretty well, so sometimes it's a little harder to kind of pop that in depending on how much feed she has in there. And so when we were talking before on kind of the fermentation vat and it being, you know, that anaerobic environment, it's going to be warm. So what Ben's holding, you know, I'm sure that feels nice and warm on a cooler night like tonight. And what you're seeing really from a digestion standpoint, you know, looks similar to what we were looking at, you know, with the TMR, right? And so this is getting mixed in everywhere within the rumen. And you can see, I can't quite see how far, but one of the things that we usually do for an activity with students, or I do in some of the labs, is we let them reach in. We let them take a look around and feel, and you can probably see some of the feed or the digestive moving up and down. And again, that's some of that rumen contraction. So, you know, 1 to 2 contractions a minute roughly, and that's moving that feed up. Typically on the lower portion of the rumen, we're going to have more of the fluid, and then we have more of a mat with some of the fiber and different things, and then we have more of the gas that's usually at the top, which is kind of what you're seeing more with some of the feed ingredients there. I don't think I mentioned it before, but one of the things that can happen with ruminants in general is they can actually bloat. And so when they have all this fermentation going on, they have a lot of methane and carbon dioxide that is in the rumen. And so in order to get rid of that, they eructate or they belch. And that's what how they get rid of those gases. And so a lot of times people always like to joke, they say that it's the farting cows causing all the problems out there, but it's mainly from them eructating or belching that they get rid of that methane and CO2. And so that's just kind of the general outlay of what we have from the rumen standpoint. And then like Dr. Luiz was explaining before, with the rumen bags that we have here, we do all sorts of different research with some of these where the researchers can come, they can put them in for a certain time point, take them out, look at those bags, look at the digestibility and everything. So if you want to go ahead and then insert, so we would kind of crumple it up and then insert that bag in. And this is one of the trickier parts, is you kind of have to get it down into some of the liquid so it can then start moving around with some of the contractions and everything else that would go on. And we might leave this, you know, in the cow for 24 hours or longer, depending. And then we would take that out and usually spray it down with cold water. And then depending on what the protocol is, you know, take some of those samples and analyze. But what you're kind of seeing here is kind of the process. You probably don't have to go ahead and put it all in, just for a general standpoint. this is what they see. So you see how this connects here? So I was saying before, that way we don't lose it in the cow. And that's typically what we do. I mean, some of our cows here at the Dairy Cattle Center, I mean, we're doing this almost every week with some of our cows that we're looking at different things, or we also collect rumen fluid. We didn't set that up tonight, but we have a little pump that we can actually put into the cow, and it's kind of a hand pump that we can get the fluid out. We use some of the thermoses like you would put your hot chocolate or your coffee in. We use that, and then we can take that fluid and we can put it through cheesecloth, and we can filter it. And then maybe I'll turn it over to Dr. Luiz here to kind of explain some of the stuff that we do in the lab with the rumen fluid and how we can do some of that in vitro research.
Yeah, absolutely. Thanks, Ben, for the demonstration, for collecting the rumen fluid earlier for us. And thanks, Eric, for the nice explanation. So from our laboratory perspective, when we have the opportunity to collect rumen fluid, when we move back to the lab, we typically have multiple flasks similar to those that we showed earlier during this presentation. And we can test different feeds, and we can learn not only digestibility, like we described to you as the difference between the nutrients that starts in the process and the nutrient that's left after a given amount of time, but we can also measure different things. For example, Eric explained to you earlier today that in the rumen, cows will ferment some of the carbohydrates into different acids, right? Acetate, propionate, butyrate. So we can collect rumen fluid from our vials or from the dairy cow when they are cannulated, and we can analyze for those acids to learn more about, do those different feed ingredients ferment into different acids? And the other question we may have is, what do those different acids mean for milk production, milk components, etc., right? So for example, we know that corn, which is very rich in starch, produce a lot of propionate in the rumen. Forages that are very rich in fiber produce a lot of acetate, right? Other type of studies that we can run is something that became very important for us nutritionists recently, is how can we mitigate the potential environmental impact of dairy cows? And I want to make a disclosure here. Dairy cows do not pollute the environment as much as people think. But another type of research we can do is we can learn about methane. Are there specific feeds that produce less methane in the rumen of a dairy cow? And the answer for that is absolutely. There are many feeds that can be used in order to mitigate methane emissions. So from a research perspective, opportunities are endless. So.
Okay, there's a couple of questions, if that's okay. And then it just adds, there was a couple of questions as it relates to, is there concern about different types of bacteria when you introduce that rumen bag, when you put samples in? So that was just a question. You one of you guys want to answer that?
Sure. So basically, the reason why we incubate those bags in the rumen is because the dairy cow rumen is populated by multiple microorganisms. A lot of those we don't even know about, right? So I don't even think we start learning about a lot of the bacteria or protozoa that are residents of the rumen. But incubating those bags in the dairy cow allow us to have a real environment containing not only the rumen fluid, but all those microbes inside the rumen fermenting that feed, so we can really learn about the digestion process. We didn't mention that, but another thing we could do is collect rumen fluid material and analyze those for bacteria population and learn about those different bacteria and protozoa population in the rumen and how they affect that. So.
That's a good question. There's another one, just, you know, in generalities, we kind of talked about that before. It doesn't hurt the animal. I guess there was a good question in here about, does the cannula get caught on gates or any facilities that might be in the barn?
Yeah, I would say as far as that goes, no, I haven't heard of too many problems with the way our gates. I mean, that's kind of how you set things up with the pens and different things. I mean, we do have a cow yard out here kind of behind us on this backside. So, I mean, certainly there's a potential for that. But the most common thing I have seen with these is either in the tie stall barn or out in the free stall for some odd reason. If you don't put, you know, the plug back in correctly and get it fitted nice, you know, that's something that could pop out, but not typically with gates, the way we have things set up and modernized with a lot of our facilities, that's not usually a problem, but good question.
They also want to know if you named the cows. They have good numbers and identifiers above them in the stall or over in the tie stalls, but that's about it, right?
Yeah, they have numbers. We don't name the cows yet, you know, but I won't lie to you. A lot of those cows that stay with our group for a lot of time, we actually treat them as part of our group. They are co-workers, and we really like them. So, you know, I won't lie to you. At a certain point, we will start giving them names, and you know, let's see if they all learn their names. So.
What compartment of those 4 compartments that we talked about does the cannula go in? So that one's been asked a couple of times. That's a great one.
Yeah, so the cannula is connected directly into the rumen. Okay, so it goes directly into the fermentation chamber. So that's where it is.
There are sometimes other places you can put cannulas. I know when I was in grad school, worked with some steers that had it in the small intestine. So kind of the beginning of the small intestine, at the end, looking at absorption in there. So you're taking fluid in different samples. So researchers do have the ability to use kind of different protocols and processes depending on what they're looking at. But I would say for here at UW, this is the most common. I really haven't heard of too many other researchers currently doing some of the different ones.
Yeah, I think we only have rumen cannulated cows at the moment in our facilities, and it's by far the most common. So, and in my opinion, the most important, but I'm biased because I work with those on a weekly basis.
So as we kind of wrap up tonight, there's lots of questions flying in, and we do like to keep it just under an hour. But before we can go back to the questions, but I guess I want you guys to just kind of cover a little bit about careers in the nutrition area. So we spent a lot of time talking about this. Feed is the most expensive input into projects, into animals, in production facilities, and in how we raise food for people. Can you just talk about how, if someone tonight was really interested in how all this works, it really sparked something in them to maybe check this out? What kind of careers, what kind of things would they have to do in terms of like subjects that they would like in school, math or science, and maybe what other things that they could maybe experience and get some, maybe some like a job experience, but not really, maybe just to shadow somebody? What would they need to do in this area of nutrition?
So I will answer the part about subjects that they have to like and they will see, and I'll have Eric discuss more about the potential careers, if that's okay with you, Eric. And so I used to teach a lot of the basic animal nutrition course, which includes a lot of students that want to be animal scientists, but sometimes not necessarily nutritionists, you know, it could be reproduction, genetics, etc. And also a lot of pre-vets, which obviously their main goal is to go to vet school. And we don't know if they want to work with small animals, equine, cows, but regardless of your career choice, nutrition is a very important concept for you to learn. To me, as a dairy nutritionist, there are a couple subjects that are very important. But remember one thing, you don't need to love all the different subjects to work with nutrition. I don't love everything about nutrition. I love certain aspects of nutrition, and I do my best to understand those things I don't particularly like the most, which I won't disclose, but there are some, okay? But from my perspective as a teacher as well as a nutritionist, the most important concepts that we teach require you to have some understanding of biology, chemistry. Believe it or not, you have to learn a little bit of math, okay? I know a lot of people want to go to a biologist career because they hate math, okay? Don't hate math. Just try to go with the flow, you know? I know that a lot of times people don't like math, but we can help you to understand that for the biology aspect of that, okay? And depending on the areas you want to engage more, you may also have to have some interest to learn more about biochemistry and physics. So if we choose some overall subjects, that's what some of the interests that I would suggest you to dig more about to make sure you understand from a nutrition perspective. So I'll let Eric discuss more about the potential career opportunities now.
Yeah, no, that was a good start on the subject matter. And I would say, you know, with my experiences dealing with farm and industry short course and college undergraduate students and students that want to go on to vet school or graduate school, there's a huge array of what opportunities there might be. And what I would say is the most common, at least in the nutrition world, is to be a nutritionist or a consultant or a feed or sales type rep is all very common. There's lots of different industries and companies and co-ops that people can work for. Some of them maybe need more of a short course or certificate or associate's degree, bachelor's degree. Whereas even on the technical side, there can be people with masters and PhDs that are more of consultants where the sales people will work with them as well. So there's a huge array of different opportunities there. But I would say, again, on the nutrition and the consultant type of side is pretty common. And there's certainly a huge market out there when we think of the amount of money that goes into feeding animals and cattle and all the ruminant species that we talk about. And so certain times people may start off in one area or think they're going to go somewhere, and then they end up going in a different path. And that's always great too. So I would say that's kind of some of the common careers that I see some of the students go out into. And then, as I was mentioning, sometimes I have a ruminant nutrition class that I teach for short course, and we try to balance some basic rations. And they sometimes say, well, this is why I pay someone to do this, because it's so complicated. So that's kind of all a part of it.
That's great. So one person, there was a question in here about, you know, what age does these cannulas be put into these dairy cows? I'm not sure if you covered that before, but what
I
age?
don't think we discussed those. Typically for our herd, the cows that are cannulated, they are usually 2 years old or later. Okay, I don't think we ever cannulated a younger animal than that, at least that I'm aware of. Okay, so that's how typically we want. Primarily because the adult animal will be easier to adapt to that, but also because for us, from a research perspective, we want to learn more about the adult animal rumen rather than the young animals.
So there's a couple of questions about why sheep can't be fed copper and other animals can. And I can give a little twist on it and you all can add to it. But yeah, sheep, that's a great question. Sheep cannot have copper, and that's really, you know, that was great you noticed that, because you need to read labels if you feed sheep, because sheep cannot metabolize copper. It really becomes, it builds up in the liver, it becomes toxic to them. And then over time, if you keep feeding them copper, then they will have a copper toxicity and then perish and not live. You guys want to add to that one?
The only thing I would like to add is that when we work with minerals, regardless of the animal we are discussing, there are a lot of potential for deficiencies and toxicities. Okay, formulating diets for minerals is very difficult because we typically feed those in a very small amount. So we need to be very, very careful on how we formulate the diets and how we feed those. So for minerals, there's a lot that we can learn in order to make sure the animals are healthy when feeding those. So.
All right, we've got about 5 minutes left, and I think we're just going to take this one question. But make sure everybody, I put it in the chat. Please fill out the survey. We need you to do that. That's this Qualtrics I just put in the chat box. If you could please do that and let us know what you thought of tonight and what other topics you'd be interested in, that would really help us out as we plan activities for you guys, because we love doing this. And who doesn't like seeing cows? And then the other is, I will show you here in a minute the screen. Just going to do this really quick, and we'll take the last question. Is next month we will be talking about feeding swine without ractopamine, and our 2 speakers are from Ohio State and Kansas State. And so Dr. Moeller and Dr. DeRushi will be talking about that. And then if you are looking for educational credits, like I had shared the link prior, the link has been in the chat numerous times this evening. If you can locate that, or I'll put it in the chat one last time. It was in your emails of confirmation. Go ahead and fill that out from this evening, and that should get you, and you can turn those into your county fair or your committees in your local area. So here is that in the chat for the verification form. So the last question that we had that I thought was a really good one. Do beef cattle belch as much as dairy cattle since their rations are different? So do beef cattle belch, as Eric was saying, as much as dairy cattle? And we'll end on that question tonight. Anybody want to hit that one?
Everything depends on the diet you feed, okay? So there's a lot of variation in what that happens. But in my opinion, I think the dairy cow is more prone to that because the dairy cow consumes more feed, consequently digests much more material and generates much more gases, okay? But regardless of the ruminant animal that we are discussing, this will happen, okay, to different extents, and the diet plays a major role.
And I guess the only thing I'll add on to that is, you know, we have the ability here with some of the green feed machine that we actually measure some of that. We look at the gases that come out depending on different diets that we're feeding. We've looked at different things with breeds of dairy cattle. So you can do the same thing on the beef cattle side, because you can put the animal up, they can eat a little bit of a concentrate or pellet out, leave them in there for 15, 20 minutes, and it captures everything like that. So that type of research, you know, is certainly out there. I just don't know some of the literature and whatnot, but we do have the capability, and we're looking at that from a greenhouse gas emission standpoint, that, you know, how can we reduce our carbon footprint in the egg animal industry?
Yeah, and the only other thing is, and it doesn't matter where the cow is, if it's a cow that lives in a free stall or on pasture, that's a natural behavior of the dairy cow. It's physiological, okay? Fermentation leads to gas production and milk. So you cannot avoid that. So if you hear any scientists say that they eliminated that 100%, you can tell them they are lying. And I will back you up, I promise. So.
You've heard it here, people. Dr. Luiz said so. So, all right. Well, thanks everybody for being on with us tonight. I'm going to flip my camera around so maybe you guys can see me tonight. But thank you for participating. Maybe we'll get the camera around. Thank you for participating. Thanks, Ben. Thanks, whatever number cow we're looking at, for showing. And thanks, Eric, and of course, Dr. Luiz, and Lori, I think is with us at Lori Wick. And yeah, so take this information and move into your projects this year. And don't hesitate to call you to Madison or myself, and I can connect you to whoever within the department that can answer your questions. So have a great night, and we'll see you next month for the Feeding Swine webinar. Take care, everyone. Good night.
Bye.
Thank you.
Good night.
Bye.
What do you
Bye.
say? Feeding.
Leave.
Bye.